Continuing our walk through anaerobes and things that have an impact on us, this next one has a much broader impact than just on us. Our colon is full of anaerobic organisms. And we each have our own particular unique mix that probably changes from day to day and shifts according to our diets, because they're basically working off of our leftovers. Is that we can't digest everything that goes through our system, but they take a lot of what's left over and process it. In some cases, to our benefit, is that we do get some digestive products from them, including vitamin K, mobilized minerals, a number of other things. They've estimated there might be well over 1,000 different species of symbionts just in our colons, just in our large intestines. But we also have symbionts all over the place, some of which are anaerobic and some of which are aerobic. Your skin has aerobic symbionts. The upper parts of your digestive system, although that hasn't really been looked into particularly. I haven't seen anything about the lungs. I won't be at all surprised to find out if we've got, we definitely have bacteria that live in our lungs that normally, that can grow out of control and cause problems if you're immunocompromised. But I don't know if they're doing anything for us. A symbiont is an organism in a trade-off. I'll do something for you, you do something for me. Is that I'll process minerals for you. I'll make you vitamin K. I will train your immune system. A number of the symbionts that live in our colons interact with immune cells right at the lining, up on the epithelium of the colon, and exchange signals that make your immune system better able to deal with bacteria, because most of these are bacteria. And possibly with viruses, is that they're still learning. This is a fairly new thing, what they call the microbiome. It started as being strictly colonic, but now they're discovering that there's so many of these. And they're also discovering that it isn't just us, that almost every animal has symbionts living in their digestive system. And also what they call commensals, things that live in you, but they don't really take anything from you, they don't really give anything to you. And we don't generally count the things that are in there just for themselves, the parasitic organisms. But those are the three levels of trade one for another. Symbiont, I get stuff from you, but I'm not bothering you. Commensal, I'm taking stuff from you that you otherwise would be using. Parasite. A lot of alcoholic beverages, the alcohol is, I think actually all alcoholic beverages, the alcohol is produced by anaerobic organisms. Sometimes bacteria in the cases of wine, sometimes funguses in the case of beer, where brewer's yeast, which is a fungus, is involved. But you basically seal them up in sealed containers with the grains or the sugars of grapes or whatever it is that you're packaging, and they take in the carbohydrates and generate ethyl alcohol and sometimes a significant amount of carbon dioxide. So champagne, beer, the bubbles come from carbon dioxide. And alcohol for other purposes, for like laboratory purposes and stuff, generally is generated. Pasteur, originally his work was in alcoholic beverages and figuring out how it all worked and what microbes were involved and what you needed to do to make them feel good and what bothered them, and then he eventually discovered, when they're bad, like in the dairy industry, we need to be able to kill them. So pasteurization arose, but also probiotics in yogurt, which is only like 3 species out of the over 1,000 that we have in our intestines. So how beneficial it is to bolster your colonic bacteria with a few from probiotic pill or from probiotic foods is iffy. Not hurtful, but iffy. It's anaerobic bacteria that make your milk go sour. As pasteurization kills the dangerous bacteria, the potentially deadly bacteria in raw milk, but the ones that make your milk go sour are a little hardier, as the pasteurization doesn't kill them unless you ultra pasteurize. Pasteurization, if you were to pump up the heat pasteurization, it changes the flavor of the milk in a way that people don't like. Ultra pasteurization uses, fancy ways to take out those other bacteria with either, flash heat, radiation in some cases. There's a number of different tricks. But it turns out that not just yogurt, an awful lot of the flavor and smell and texture of cheeses comes from particular types of anaerobic organisms. Brewer's yeast is related to baking yeast. They both produce ethyl alcohol and carbon dioxide. Is that baking yeast doesn't produce a lot of ethyl alcohol, and the way that it produces it tends to volatilize. It evaporates away really quickly. I mean, it definitely does when you bake stuff. But they're primarily for the bubbles of carbon dioxide gas that they produce, which makes your dough rise. As the guy on the British baking show says, when you put your yeast in, don't throw the salt in on top of it and mix it up with the water, because the salt will kill your yeast. We'll talk about that when we talk about diffusion and osmosis. You put your yeast in on one side and your salt in on the other, and then you mix them together so the salt levels around the yeast never rises to the point where osmosis causes water to leave them and it kills them. So, but baking is pretty important. A lot of times in things like cheeses, the flavors come from various types of acids, lactic acid, propionic acid, other types of acids that are generated by the bacteria. Lactic acid generation is what makes your milk go sour, is what will eventually drop the pH to the point where the fat will curdle out of it. But that's also used in the production of curds and cottage cheese and other kinds of things. As far as human impact is concerned, I guess that's pretty much it. We talk about the process of aerobic respiration. Well, the process starts with anaerobic. So you start with glucose and oxygen. It's a whole bunch of enzymes and such involved. Little squiggly arrow is kind of it isn't a product, but it's an offshoot of the process. So it looks like photosynthesis backwards. As you start with these in photosynthesis and wind up with these. Here you're going the other way around. And from a distance, the steps look the same in reverse. Is that photosynthesis starts with water coming in an electron transport chain, and then goes into a cycle, and then produces half molecules of glucose that are combined into glucose. Aerobic respiration takes the glucose and breaks it in half, feeds it into a cycle. The cycle produces a whole bunch of ATP through an electron or feeds into an electron transport chain that produces a whole bunch of ATPs. And ultimately, the oxygen picks up hydrogen, you wind up with water at the end, as opposed to water going in at the beginning. Now, obviously, the steps are not going to be using the same enzyme, so it doesn't work exactly in reverse. You have to take a step back to see the big stages are kind of a reverse of each other. But it's still kind of cool that one goes forward and one goes backwards. The anaerobic stage, in eukaryotic cells, the anaerobic stage happens in the cytoplasm. The assumption is this is what our distant ancestors did before they kidnapped aerobic bacteria that could take the products of our anaerobic respiration and further break it down and get more ATP from it, doing it. So our mitochondria are specialty areas that actually use the oxygen. Our cytoplasm has the chemistry that just breaks the sugar apart. Glycolysis breaks the sugar in half. Now, I'm going to walk you through the steps, and because I want to have an overhead here, I'm not going to write all this stuff down. I'm expecting you to kind of write the stuff down as we go through it. When potato salad goes bad, that is anaerobic respiration and not little things of potato salad with guns. I don't think. This can happen in our cells. We'll talk about that eventually. Primarily, we wind up here with pyruvic acid. We can also wind up with lactic acid. This down here is the production of ethyl alcohol. So there's very similar kinds of processes. Okay, it's kind of the bare bones of this that I'm going to expect you to know. So you got glucose. 6 carbons, but one of the carbons is not on the ring. It's a 5-carbon ring with that oxygen bridge, which you always get in carbohydrates, and 1 carbon loose. ATP comes in. You have to invest some energy to get some stuff out. You have to invest ATPs in order to get more ATPs ultimately. So it uses ATP and group transfer potential. It sticks the ATP phosphate on that extra carbon. Shakes the molecule rearranges. It converts from a 5-carbon ring with 1 carbon sticking off to a 4-carbon ring with a 2nd carbon sticking off. Then ATP comes in and tags another carbon with a phosphate. Not easy to get the phosphates onto the ring carbons, but it's easier to get them onto the extra, disassociated. No word I can come up with actually works right. It puts it there. Again, destabilizes the hell out of the molecule. Now it breaks the ring. You wind up with a 3-carbon molecule and another 3-carbon molecule. So there's 2 of these. This particular progression doesn't show them as pairs. So you have to kind of keep in mind from here you're dealing with 2 of these. Now, this molecule is unstable enough that it can grab a phosphate right out of the juice. So it doesn't need ATP to sacrifice a phosphate to it. It doesn't need to break ATP down to do this. It can grab a phosphate itself. And so you wind up with the phosphate that was put on there and the phosphate that it grabs. Each one of these can now use the phosphates to make ATP. So you invested 2, it grabbed 2 more, you're going to get 4 back. You invested 2, you're going to get 4. Not a huge payback, but you're doubling your input. And it goes through some steps here to do that, where you can see ATPs coming out and ATPs coming out, water coming out. What you're going to wind up with is pyruvic acid, which is your 3-carbon molecule. Now, there's been a push in chemistry to take the ic acid designation away and make them 8s. So pyruvate, and lactate. Lactic acid, lactate, same thing. Pyruvic acid, pyruvate, same thing. This is not quite what's going to be fed into the aerobic system, but it's close. So, steps. You got a glucose. ATP sticks a phosphate on the non-ring carbon. Screws up the molecule, you wind up with another non-ring carbon, ATP sticks another phosphate in there, breaks the glucose into 2 single phosphated molecules that each grab another phosphate out of the juice. Each one of those phosphates is now capable of being used to make ATP. So you put 2 in, now you get 4 out. Now you've got half a glucose molecule. I'm sure you probably can't read this, so I didn't want to put it in here too early. ATP's in, ATP's in. You also get NADHs out. Here's an ATP payoff and another ATP payoff. Here's water coming out. And you wind up with 2 pyruvates. So here's glucose, here's glycolysis, here's ATP being generated, here's NADH being generated, here's pyruvate being generated. Now pyruvate is going to pair up, is going to stick to acetyl coenzyme A. Sometimes pyruvate is processed into lactate. It's weird that they've got the 8 here and not there. Now you've got your cycle inside your mitochondria. See, mitochondria. Oxygen's going to come into this, combined with hydrogens to give you water. Carbon dioxide is going to come out of this cycle. NADH is going to come out of this cycle, feed through the electron transport chain to produce ATP, and eventually tag the oxygen with hydrogens, which are carrying the electrons. Okay, let's go back here. The aerobic stage is in mitochondria. And let's see what we got here. Hydrogens come peeling off of a lot of these molecules and help to produce NADH and FADH2, which are electron carriers. And the electron carriers are going to feed hydrogens through the electron transport chain and produce ATPs. So you get a bunch of them, you get a bunch of them, you get a bunch of them. You'll see this all feeds into the electron transport chain. You do wind up with a couple more ATPs This is the weird molecule that the coenzyme winds up being actually replacing one of the carbons on the pyruvate. I'm trying to leave some of these weird details out, but I can't help myself. I have to mention them. Now notice here. So what are you getting at the end? Well, you're getting 2 ATPs from the anaerobic stage. You're getting about 34 from and 2 here from the aerobic stage. So you're getting about 38. These numbers vary depending on the source. Since the yield is indirect, it's hydrogens going through a membrane carrying electrons until the electrons are kind of depleted, but the electrons can go through more than once. So you can't say, okay, this NADH is going to give us like 4 ATPs. You're not, is that you can calculate roughly how many ATPs you get. I actually have it written down here somewhere. The NADH yields about 2.5 ATPs. The FADH2 about 1.5 ATPs each. So there's actually GTP in there as well. It gets a little crazy. I'm not going to expect you to remember this at all. I'm just going to show it to you. You've got a 4-carbon molecule here. Remember, 2 carbons and acetyl coenzyme A are coming in, and you get a 6-carbon molecule. Except it's not glucose, it's citric acid. It rearranges, it oxidizes and generates NADH and a hydrogen ion. 1 carbon dioxide peels off of it. Now it's 5 carbons. Another carbon dioxide peels off of it. It's a 4-carbon molecule. It's an energized 4-carbon molecule. It can be used indirectly to make an ATP. It can be used to generate FADH2. Water can feed into it and feed off more hydrogens. So there's a whole bunch of stuff coming out of here. But what you're mostly getting are NADH and FADH2. And you're breaking, you fed in a 2-carbon molecule and you get 2 carbon dioxides out. This is where the carbon dioxides come from. Again, the electron carriers is the big part here. So this is the Krebs or the citric acid cycle, or the citrate cycle, although to tell you the truth, I've never heard anybody call it that. And the electron transport chain. So roughly it generates 36 ATPs. So it's about 18 times more efficient than the anaerobic stage, which only generates 2. But it's not precise. And there are situations where in 1 minute it may be generating more than another. Is the electrons don't all necessarily tire out the same amount. The membranes don't always process the same amount. But that's it, briefly. If you wind up getting into biochemistry, you'll wind up going into this more. If you get into any sort of physiology, like I talked about my plant physiology class, you'll get into, I don't remember getting into this a lot in the plant physiology class, but the photosynthesis was equally crazy. Okay, is that we've got other things that can feed into this cycle that we'll talk about in the next video.